The title of this work is Steady-State Imaging with Inhomogeneous Magnetization Transfer Contrast using Multiband RF Pulses. The authors are Shaihan Malik, that's me, Rui Teixeira, Daniel West, Toby Wood, and Joe Haynor, and all of us are from King's College London. So this work actually evolved as an extension to a previous piece of work led by Rui Teixeira. So I'm going to first of all talk a little bit about that work before going on to discuss the Inhomogeneous MT effect and how we measured it using steady-state sequences, which was the subject of this work. So Rui was looking into variable flip angle relaxometry, measurements of T1 and T2 using balanced and spoiled gradient echo sequences with different flip angles. And he found that magnetization transfer effects were a big confound for those measurements. By magnetization transfer, I refer to transfer of magnetization between the free liquid water that we are normally imaging with MRI and magnetization in semi-solids that exist as a background in most biological tissue. The issue is that semi-solids the magnetization is saturated when you apply an RF pulse. That magnetization can transfer with the water and it can lead to a difference in the measured signal from the water. So the problem for variable flip angle sequences is that when you change the flip angle to try to measure the T1 and T2, you also end up changing the RF power because you either have to change the RF pulse's amplitude or duration to do that. And the saturation of the semi-solid is dependent on the RF power. So by changing the RF power, you change that saturation effect, and you're in effect changing more than one variable at the same time when trying to make your measurement. This is illustrated by the cartoon shown here. This is a Gaussian RF pulse being scaled up and down to indicate a flip angle being scaled up and down. And on the right is a graph showing the RF power as a function of the flip angle, and it goes quadratically in this case. So if you were trying to measure T1 and T2 by looking at different flip angles, you would also be making measurements which use different levels of RF power, and that leads to inconsistent results. The solution to this proposed by Rui was to employ multiband RF pulses, such as the ones shown in the bottom cartoon. Now, these types of pulses are usually used for simultaneous multi-slice imaging at the moment, but in this case, there was no underlying slice select gradient. This was non-selective imaging. Instead, the additional bands shown in red are placed at a high frequency offset from the water resonance, so they don't do any excitation, but they do saturate the magnetization in the semi-solid. The idea is that if you are scaling the flip angle up and down, and the flip angle depends on the power in the on-resonant band shown in green, you can compensate by adding or taking away power in the off-resonant bands such that the total power is constant. You see on the graph on the right, for the multiband pulses, the RF power can be held constant no matter what the flip angle is. And what was found was that using these types of pulse led to much more reproducible measurements of T1 and T2 using rapid steady-state sequences. And you can read more about it in the work shown here. Now, at around the same time, people were describing an inhomogeneous magnetization transfer effect, which could be used to probe tissues like myelin, which display a strong ihMT effect. So the ihMT effect is essentially saying that there is a difference now in how the system interacts under different types of off-resonant irradiation. So in the cartoon here, you can see the classic type of MT experiment where you apply a certain amount of saturation, shown as W, off resonance. So the free water resonance is shown in green, which is very narrow, and the semi-solid is in blue, and it's quite broad in frequency. So if we saturate off resonance, we can then saturate the semi-solids, we get some transfer of magnetization between the two, and we can measure a different signal in the free water. You can do the same thing by applying the same total amount of power, but spaced equally around the resonance, so shown as half the power at positive and negative frequencies. And using the sort of classic MT picture, you would expect these two scenarios to lead to the same measurement. However, it's been found that you actually see a difference between these two. And this is not due to an asymmetry in the blue absorption spectrum. It's actually due to non-motion average dipolar coupling occurring in the semi-solid. And this has been shown to correlate quite well with myelinated tissues in the brain. So the idea now is that the semi-solid contains not only the Zeeman magnetization, which is the what we usually measure in MRI, but also some dipolar ordered magnetization due to longer lasting dipolar interactions between neighboring molecules. And you can cause a transfer of magnetization between these two compartments using off-resonant RF pulses, which I'll describe in a minute. Now, current ihMT protocols then try and bring out this difference by applying either the dual frequency saturation or a single frequency saturation, as shown in the graph above, or in the diagram above, followed by an imaging readout to then compare the two conditions. So what we noticed was that the types of sequence that we proposed for our controlled saturation work that I mentioned earlier on were also conducive to making this type of measurement. So the diagram here shows a balanced SSFP scan with a multiband pulse excitation, and this multiband pulse could be have a two-band pulse, so a single band on resonance for water shown in green, and then some off-resonance saturation shown in red or pink. Or we could have dual frequency saturation, where we split the amount of off-resonant saturation equally between plus and minus the same frequency. And these two should lead to the same effect in a classic MT picture, but different effects in our ihMT picture. So the basis of the work that I'm talking about now was to develop the signal model to try and understand how we would expect steady state sequences to respond when faced with these two different types of excitation, and then to investigate the use of those sequences for imaging with ihMTcontrast. A large part of the work was then based on development of that signal model, and you can read about that in the paper if you're interested in more detail. Just to give a pictorial overview of it, I'll show you now a diagram. So let's say that we have a sample that contains only free water. This is then governed by the Bloch equation, so you can write down your magnetization vector, mx, my, mz, and the dynamics of this vector are governed by the Bloch equation. The classic model for most tissues, which works quite well, is known as the binary spin bath, or this two-pole MT model, where we add a semi-solid component here, which has some Zeeman-ordered magnetization shown in blue, and this magnetization exchanges with the Z component of the free water magnetization, and the interaction between the semi-solid magnetization and our pulse sequences is occurring when we apply RF pulses. We saturate that part of the magnetization. This is used extensively for quantitative MT and other types of measurement like that. A more complicated model, and this diagram is picking up on a model proposed by Gopal Varma with the citation shown at the bottom of the slide, has now a two-compartment model. We have the semi-solid itself consists of two compartments. One of them is the classic Zeeman-ordered magnetization. The other one consists of Zeeman-ordered magnetization, which is coupled to the dipolar-ordered magnetization. And these latter two components exchange with each other, but only under off-resonant RF irradiation. So you have to apply off-resonance RF to make those two exchange with each other. The coupling is linear in the offset frequency, which means that if you apply power equally at positive and negative frequency, the terms cancel and you get rid of the coupling between those two poles. And that is how you can then obtain this inhomogeneous MT contrast. So we describe in the paper then a matrix-based signal model that allows us to compute signals that we would expect from balanced and spoiled sequences. And the conclusion from that is that you can, if you look at the way the matrices have been redefined, you can then use those in well-known expressions for steady states for balanced and spoiled sequences, but replacing the matrices that you would have from your normal Bloch equation model with the more complex model instead. So in this work, we've used 4 different types of RF pulse, single band pulses, which are just on resonance only. We don't expect these to cause any coupling between those two Zeeman and dipolar poles because there's no off-resonance RF being applied. 2-band pulses, where the off-resonant bands, shown in pink, can either be at a negative or a positive frequency, and these we do expect to generate this coupling between the Zeeman and dipolar orders. And then a 3-band RF pulse, which has equal power split at positive and negative offset frequencies, and we don't expect this to have any coupling because those terms cancel out. So in order to investigate this, to validate our signal model, we used a classic MRI physics phantom. This is a lunchbox full of water with some sample tubes inside. This was imaged on 1.5T Philips system using balanced and spoiled sequences with a range of flip angles and both types of sequence, and also a range of different pulses, so single, double and triple band pulses. And we fitted all of the signals together to our signal model to see how well a model can describe the data. So shown here is the first sample, which was water. This is a control, showing that in this case you can fairly well describe the curves only with a T1 and a T2 and an overall amplitude. And this is because this really doesn't have any MT effect. So the different types of multiband pulse don't affect the water sample differently from each other. On the other hand, BSA, that's bovine serum albumin, which has been chemically cross-linked, you can see here there's a strong difference between the single band pulses and the multiband pulses, because the multiband pulses have a stronger amount of RF saturation. The model fits the data very well. We don't see any difference between 2 and 3 bands. That's because there is no inhomogeneous MT effect displayed in this phantom. And the parameters that give rise to the model fits shown in the table are all quite plausible for this sample. Now, on the other hand, PL161, that's prolipid 161, is an ihMT phantom. It was first proposed for this use by Swanson et al. In the reference shown below. This substance does have a strong inhomogeneous MT effect, and it can be seen from the graph because the 2-band pulses, so the 2 plus and 2 minus, shown in yellow and orange, cause the signal to be rather different than the 3-band pulses, which are in purple. And that difference is the ihMT effect, which we were trying to measure. So you can see that the effect is there, and that the signal model can fit those curves very well. Finally, other people have used hair conditioner as an ihMT phantom. There's a mention of it in Varma et al. Shown at the bottom. Other people have also used it. And again, you can see a strong difference between the 2 and the 3-band excitations, which is the essence of the ihMT effect. So we also optimized our sequence to give strong ihMT contrast in vivo. And this is the result of then a subsequent in vivo experiment using a 30-degree flip angle with balanced SSFP, using 1, 2, and 3-band RF pulses. The ihMT ratio is the difference between the 2 and the 3-band excitation divided by the single band. And all of this was steady-state 3D imaging, so we obtained multiple imaging volumes, registered them together, and then looked at the differences to make these ratios. The MT ratio shown below is more like the classic MT ratio, so that's the difference between multiband and single band, again normalized to the single band. What you can see is that the ihMT ratio and MT ratio look rather different to each other, and that the ihMT ratio appears to be more white matter weighted, which is what we would expect from ihMT contrast. So to conclude, we developed this matrix model that allows signal predictions for rapid sequences, and it gave very good fits to phantom data. The in vivo images are consistent with our predictions, and we did achieve ihMT contrast. The numerical model could be used subsequently in the future for quantitative uses of the same types of sequence if we want to try and estimate these parameters. So just a note on reproducible research. This is something that we're very keen on. This is a screenshot from the paper. If we zoom in, we see that we've referenced a GitHub repo. We published the code and the phantom data as well, so everything that's needed to reproduce most of the results in the paper simultaneously with publication of the paper. This is a screenshot from the GitHub site. So we've got the GitHub handle MRI Physics, and on that site there are a lot of repos. Each repo corresponds to a different publication, which hopefully will allow you to replicate the results if you would like. And we've also provided a README file which you can look at for documentation if you would like to get more help in understanding how the code works. If anybody is interested in trying it, please take a look, and we would welcome any feedback about that to see if, to spot some mistakes, but also to see if there's anything that could be improved. So thank you very much for listening. I'd just like to say thank you to everybody in the group who contributed to the work, and also to Olivier Giraud, Guillaume Duhamel, and Gopal Varma for their useful advice and discussions relating to this work. So thank you very much.